If you're a lineman in charge of keeping the lights on, Grainger understands that you go to great lengths and sometimes heights to ensure the power is always flowing, which is why you can count on Grainger for professional-grade products and next-day delivery, so you have everything you need to get the job done. Call 1-800-GRAINGER, click Grainger.com, or just stop by. Grainger, for the ones who get it done.
Hello, everyone, and welcome to Talk Nerdy. Today is Monday, May 26th, 2025, and I'm the host of your show, Dr. Cara Santa Maria. And as always, before we dive into this week's episode, I want to thank those of you who make Talk Nerdy possible. As of right now, I think Patreon, which is the platform that we use to help support the show, has been working really hard in negotiations with Apple. And as of right now, there have been no changes to the way that you sign up for the show or the way that you pledge your support. Although we do still recommend that if you are using an Apple device, if you decide to sign up to Patreon, that you do it through their website and not through an app from the Apple Store, because I think there's still some confusion about whether or not Apple is taking a pretty large cut. So highly recommend you use the website. If you're going to do that, it's patreon.com/talknerdy, where you can learn more about how to support the show on a per episode basis. This week's top supporters include Anu Bhadavaj, Daniel Lang, David J.E. Smith, Mary Neva, Brian Holden, David Compton, Gabrielle F. Jaramillo, Joe Wilkinson, Joseph Lamore, Pasquale Gelati, Rika Maharaj, and Ulrika Hagman. All right, let's just dive right into it. So I had the opportunity to chat with Dr. Maggie M. Fink. She's an adjunct professor at Indiana University South Bend. She's also a postdoctoral researcher at the University of Notre Dame, or Notre Dame. I don't know how you want to pronounce it. She studies both science communication, or does both science communication and studies bacterial genetics. She's also an artist and a poet. She co-hosts the podcast Rust Belt Science, and she recently co-authored the new book, The Color of North, The Molecular Language of Proteins and the Future of Life, with her co-author, Shahir Risk. So we're going to dive into both SciComm and proteins, which sound like two very different things, but I promise you there's a lot of crossover. So without any further ado, here she is, Dr. Maggie M. Fink. Well, Maggie, thank you so much for joining me today.
Thank you so much for having me. I'm very excited to be here.
Yeah, I'm excited to talk about your work and to talk about your new book that you co-authored with, let's see if I can pronounce his name. Is it Shahir Risk?
Yes, Dr. Shahir Risk. Very good.
Oh, look at that. Oh, first try. So the book is The Color of North, The Molecular Language of Proteins and the Future of Life. So there's a lot to unpack, even just in the title. But before we get into that, I would love to learn a little bit more about you, how you came to be interested in this specific topic and related topics. So are you actively working on your postdoc right now?
Yes, I'm actively a postdoctoral researcher.
Awesome. And how long? Because I know for me, I did a PhD in psychology. The postdoc, which we also sometimes call the fellowship because it's both clinical and research oriented typically, is 1 year, right? We do a pre-doctoral internship and then we do a postdoctoral fellowship and then we go out for licensure. But I know a lot of friends who do research degrees in the sciences where their postdocs can go on and on and on. So I'm curious, where are you in your postdoctoral studies and what do they look like for you?
Yeah, I am about to finish my first year in my postdoc position, actually. I defended my thesis last summer, so I'm still kind of in the thick of adjusting to postdoc life. I did my PhD at the University of Notre Dame, where I'm still a postdoc. I switched labs, but I'm still at the same institution, which is a bit of a unique thing. Most people switch institutions. But my postdoc is also a little bit unique in that my time is actually split 50-50. Half time, I work in a lab doing bench work, really traditional postdoc for a microbiologist. And the other 50% of my time, I'm actually working for the College of Science here at Notre Dame in the dean's office as a science communication postdoc. So I approached for this role a little bit over a year ago, and said I really want to continue to do traditional academic research as well. So that's how we kind of came up with this hybrid model for me to be able to do the science communication work that I'm really passionate about, as well as continue on the research that I also love because I'm a scientist. So yeah.
I love that. And so what does SciComm look like for you? So you have a podcast that you co-host, Rust Belt Science. You obviously co-authored this book. What different avenues do you find that you enjoy doing science communication in?
The book, obviously, writing and doing that type of stuff, I really have enjoyed over the past several years working on this book. And particularly, this book has artwork in it as well that I did with my co-author. So I really like science communication at the intersection of science and the arts, and how do we use art to effectively communicate science. But I'm also really passionate about connecting with the community that your institution is a part of. So here at Notre Dame, the city of South Bend, which is where I grew up and was raised, it's really important for me to find ways to connect to the local community who's directly impacted by the work that the university is doing on a day-to-day basis, and then a lot of the research here impacts families. So finding ways to get scientists, graduate students, postdocs, professors off campus into the community, doing citizenship science. What does the community need and want from us, and how can we build that bridge, build that trust back with my community, and then how do we teach scientists how to go to their own communities wherever they end up and do that. So I do the science communication, and then I'm also involved in training other scientists as well how to get out there and do that.
I love that. And when it comes to SciComm, very often we are communicating our own science, sure, but we're also, like you mentioned, helping with SciComm training, helping with translating scientific concepts to the public. There's a broader umbrella very often as science communicators of the types of sciences that we tend to communicate. But I'd love to maybe switch gears a little bit and talk more narrowly about your science specifically and where your passion lies. What did you study in undergrad? What did you was it a big shift when you went to grad school? How did you kind of discover your area of science?
Yeah, absolutely. I mean, every scientist will talk about their science forever, so it's an easy thing to talk to people about. As an undergraduate, I was a biochemistry major, so I did a lot of research with protein engineering and
Mhm.
designing new proteins to do new things. So these proteins are these molecules that do all these jobs inside of our bodies, and we can actually take them and change them, just make really small changes, and they can then do something new and different. So in the case of the lab I worked in, we actually took these biological molecules, these proteins, and engineered them to bind and grab onto glyphosate, which is the active ingredient in the herbicide Roundup.
Mhm.
So we want to be able to detect that in soil, so we can use these proteins to detect those. They give off a fluorescence. So we can see with light, and we can measure if there is a contamination in the soil. So that's the type of work I did as an undergraduate. Really fell in love with biochemistry and proteins. That's what this book is about. When I went to graduate school, I thought for sure that's what I would continue on in. I was rotating in labs that did protein engineering for like immunotherapy for cancer, and antibiotic resistance and things like that. But in my program, we all had to do one biology rotation in addition to whatever else we were doing, in my case, biochemistry. So I called up this professor and said, "Hey, can I rotate in your lab? I know you're a microbiologist. I'm not really interested, but I have to do this." He said, "Sure, you can come spend 12 weeks here, and we'll maybe we can use some of your biochemistry expertise in our lab, and you can help out there." And within the first week, I immediately abandoned all hopes of being a protein engineer and fell absolutely in love with bacteria, microbiology, and in particular the work that was being done in my advisor's lab, Dr. Joshua Shrout, because he approached microbiology from a sociological lens. So, how do bacteria behave? How do they interact with their neighbors? How do they share food? How do they fight off enemies? And asking kind of these interesting, the sociological questions, and then we can use traditional microbiology to answer them. So I was hooked. I studied a bacterium called Pseudomonas aeruginosa, which is really common in lung infections, and it's a really pretty blue green color. So yeah, I kind of made the switch from proteins to bacteria. And now as a postdoc, I am working on tuberculosis and understanding how that bacteria is able to infect and cause such damage in hosts. So microbiologist now.
It's an important time to be studying TB. I mean, you wouldn't think it would have become an important time to be studying TB, but it is, and we often think of some of these infections as infections of old, although they do still globally affect so many people, but it's even seeing a recurrence here in the US. Um,
Yeah, and TB, it affects a lot of marginalized populations as well. It's really common in prisons. There's a lot of TB outbreaks there. So yeah, you kind of think of it as maybe in underdeveloped countries this shows up, but it shows up in the United States in a lot of marginalized communities as well, and it's very difficult to treat. So yeah, I'm very privileged to get to work on it and hopefully contribute to the field in some way at this point. Yeah.
It's always interesting to me, and I think that this probably is relevant for the book as well, but it's interesting to me how there are these different paths to answer important questions using scientific methodology. So very often in an academic setting, somebody will learn a technique or they will learn an approach through their laboratory training, and then they'll discover that that approach or technique can be used in a bunch of different ways. And then, okay, now all of a sudden I'm answering questions about, like you mentioned, immunotherapy, when before I was answering questions that were relevant to agriculture. And then you've got the flip side where you've got individuals who are like, I'm really passionate about this like social justice issue or this specific health question. And now I want to figure out all the things I need to learn in order to answer that question. When we talk about proteins, I mean, proteins are everything,
Yes.
right?
Yes.
Like, yeah, there's a million different things that you can do if you really understand the nature of proteins and how to manipulate them and how to, I don't know, harness their
Yeah.
power, I guess
Great
you could
word.
say.
Yeah.
So let's take a step back and let's even just talk about the title of the book, because I think to most individuals, The Color of North, first of all, they're like, that makes no sense. And second of all, they're like, what does that have to do with protein? So I'd love to know how you guys landed just even on that title.
The title went through many iterations, and this was the final one, The Color of North. And we did want it to be a little bit intriguing and not something that would immediately off-put somebody to like, oh, this is going to be too sciencey for me, right? To maybe draw on some interest. And on the cover, if you look at our book, there's a bird, and that corresponds to the title as well, because birds actually can see, I'm using air quotes here, but see the magnetic field. They can see north somehow. We don't know what it looks like to them, but they have these proteins in their eyes that can actually detect the magnetic field and course and translate that into some type of signal that then these birds can use for their migratory paths. So it's one of those mysteries in the natural world of how they do this, but we do know that it is a specific protein that many different types of migratory birds have that they can detect. So that's kind of the science behind it. And we thought it was a kind of a more poetic title for a book that's kind of might be a little bit of a scary topic for people thinking about chemistry and things like that.
Yeah, that makes sense. Oh, I love that. And so the molecular language of proteins and the future of life. But before we talk about the future, we need to understand the present and the past. Let's just start very, very simply. I mean, deceptively simply. What is a protein?
Yeah. Most people think about proteins as food. That's the first thing that pops into
Mhm.
people's mind.
Oh, you're right. That's
Yeah,
funny. Yeah, most people think, there's sugars, there's protein. Yeah.
and
Yeah.
food, they're not incorrect. Food is made up of proteins. The same proteins that are in our food are in our bodies, are in the bacteria. So yes, it's food, but it's also everything else that biology does. So a lot of people also are very familiar with the idea of DNA, the genetic code. Everything that tells our bodies who we are is in our DNA. And it's very important. And there's been so much research on it. It's iconic. Everyone understands that. They even might know to some extent what a gene is, right? You have a gene for your eye color, or this gene might correspond to a disease. But all that DNA actually is the instructions for how to make a protein. So the DNA is like your IKEA manual, and then there's all these machines in your body that read it and then build the specific protein that needs to be made to carry out a function. So you might need an enzyme, that's a type of protein that breaks down food in your body. So your body will find the instructions on the DNA for how to make it when it needs it and put it together. And there are these really complex three-dimensional structures. DNA, we recognize that helix structure, but proteins have all types of different 3D structures that we've actually been able to figure out. So yes, we need DNA, but the proteins are the things that are actually doing the jobs inside of our bodies.
Yeah. And so when we think about, I mean, tell me, gosh, I did a master's in biology with an emphasis in neuroscience back in 2007. And, because my background was psychology before that, it was like a huge shift and a big education and just like, this is a cell and this is where proteins are made and this is biological macromolecules. And I used to teach like intro bio. And I would always think in terms of like, there are these macro molecules. You've got proteins, you've got sugars, you've got fats, and you've got nucleic acids. And these are like the four kind of big categories of like the stuff in your body that does stuff. But then proteins was always like, it was like, okay, let's talk about DNA and RNA. Let's talk about lipids. Let's talk about sugars. Now we're going to talk about proteins the rest of the time.
Right.
Because they do everything. They're so complex. They're so complicated even in their structure because there's these multiple levels of structure. You can break them down so many different ways.
So many proteins that we have in the human body also exist in birds, plants, fish, basically every living thing. We can find a similar protein or maybe even an identical protein. But then the interesting proteins come when you start to have these really specific things that an organism might do, where they have a protein that's completely unique to them that they evolved to use for maybe seeing, the magnetic field or bioluminescing in the ocean. So there's so much similarity between us and the bacteria that's in your gut. But then as you start to branch away, the proteins become more and more wild, and the things that they can do, the chemistry that they can do, is just fascinating.
I love it. And so anything specific when you first kind of discovered how, I don't know, multifaceted or how interesting proteins could be, anything that you were really drawn to that said, oh God, I got to know more about this specific function or this specific application?
I do remember sitting in my intro to bio class, and I can't remember the exact number now off the top of my head, but just how many possible proteins there could be in the world. Like if you take the combination of what you could make, it's just astronomical how diverse proteins could be. Now, nature has not explored all of those. They've kind of repurposed the same idea over and over. But the potential for what proteins could do was kind of one of the first things that drew me to proteins, but then also the field of protein engineering and how we can take what nature has and try out some of these things that nature has never tried before. We can change the DNA to make a new protein that has this new function. And I think that the possibility and the exploration for that, and not only can you make something new, but in the process, you can better understand how proteins and the chemistry behind them works. There's so much we don't know. So you can accomplish two things at once in this field of protein engineering. And it just seemed like an endless playground to be able to do and take some basic proteins that a bacteria makes and make something completely different. You never run out of things to study and explore, and that was really appealing to me.
So let's talk a little more about protein engineering. Like I guess maybe break it down a little bit. Like what even is it to engineer a protein? Like how does one even go about doing that?
It is surprisingly straightforward to do it, turns out. Like I said, DNA is the instructions for how to make a protein. And for quite a while now, scientists have figured out how to go and change DNA. And we do this using E. coli, a bacteria. We can go in and manipulate E. coli DNA and change it from saying, okay, instead of adding this molecule, add this molecule. We're going to change, some of these molecules that are on the protein. And then E. coli will make that protein for us because it knows how to make proteins. It needs to make its own proteins. So we can have E. coli make this new protein for us, and then we can test it. And depending on what kind of function we want to do. And so that's actually, you can have a brand new protein in about a week once you get that DNA change. So it's pretty easy, pretty inexpensive to do as well. And scientists do it all the time, even if they're not designing the next miraculous protein. It's something that's really it's a common lab tool that we use to genetically modify these E. coli to make proteins for us. That part is straightforward. It's pretty straightforward. It's a really standard lab protocol that people follow. But the tricky part, and the part that people get their PhDs in, is really understanding the chemistry behind these proteins. Because proteins, like I said before, have these very complex three-dimensional structures. And that structure is integral to their function. So we can look at a structure of a protein and sometimes get a pretty good guess what it does just by what we know it looks like. So you have to be able to maintain the structure that you want when you're making these changes and still get the change that you're interested in without disrupting too much. So there's a lot of understanding of chemistry that goes into this field of protein engineering. And actually, this past fall for the 2024 Nobel Prize, one of those went to one of the first people who engineered a new protein. It's a really growing field and has been for the past few decades, and it's really big in medicine and drug treatments. Now looking at proteins as a way to treat things like cancer or autoimmune diseases. So now they're on top of those medical purposes. A lot of scientists are exploring environmental ways to use these engineered proteins.
And when we talk about an engineered protein, I mean, as you mentioned, there's the actual kind of protocol or the logistical side of engineering the protein, but there's also like the creative side of designing the protein or discovering new uses or new binding sites or all these different things. Like how does somebody even do that? Is it different now with tools like AI, like, computational chemistry sort of software programs and things? Like, are there things we can do now that we couldn't even dreamed of doing in the past?
Oh, absolutely. I think AI and a lot of the computational stuff, which is not my training at all. I haven't done much of the computational side of things. It's going to revolutionize how we do this, and I think it already has in a lot of ways. Again, Nobel Prize this past year went to AlphaFold, which is this incredible program that can predict the structure of a protein, which has just changed the field entirely. So that allows us to visualize proteins that we've never been able to visualize, even if it's not 100% accurate. And again, because that structure is so important, the ability to visualize them and to tinker with things and see how the chemistry might change before we actually go in and test it in a lab is going to speed up a lot of the process. But I think your point of the creativity behind it, the AI cannot generate anything new. So they might help you test to see if your idea is correct, but you have to have the idea first. You have to have the crazy creative idea of what if we tried this? And then those tools can help you. And the field of protein engineering, and I think biochemistry in general, is highly visual when you're thinking about proteins. All we do all day is look at protein structures and turn them around in space. And so that creative side is also a huge part of why I was drawn to proteins and protein engineering, because I think science is a creative endeavor, no matter what field of science you're in. And I think it's important that we don't lose that just because we have really incredible tools that can make the job a little bit easier.
Oh, absolutely. And I wonder if we should even just take a moment to really sit with, I mean, this is the exact wrong medium to do this because it's audio only, right? And we're talking about something that's highly visual. But if we
Yeah.
just took a moment to sort of describe or discuss the what we're talking about when we talk about the visual or the physical, the structural complexity of a protein. Can you share with us a little bit about the like levels of protein organization and all that good stuff?
Yeah, absolutely. Well, my co-author, who I don't know if I should give him more credit. He always describes protein structures as abstract art when you look at them. There are these twists and turns. There's these helices that form. So it has this really flowy, abstract nature to them. But we kind of, the sciencey way we talk about proteins is we have our primary structure. So that's just the chemicals, the molecules that make up the protein. And these are called amino acids, and there's 20 of them. So there's 20 different options when you're putting together a protein, basically. And that's the sequence. Just like DNA has a sequence of nucleotides, proteins has an amino acid sequence. And from there, as these molecules get added on, as these amino acids get added together, the chemistry starts to maybe pull some together, or maybe they want to oppose, so it'll push towards opposite ends in space. And all of these little chemical interactions happening begin to form the structure. And at that point, we have kind of two basic structures that show up in proteins. One's called the alpha helix, and that's very similar to the DNA helix that we are all a little bit more familiar with. And they can be a bunch of them all bundled up together. They could be maybe just one or two kind of throughout the whole structure. And then there's something called beta sheets, which is one of my favorite parts of proteins personally. They look like arrows when we represent them, but they're these kind of directional flat sheets that form, and they can kind of interweave together to make like basket structures. They can form these really crazy complex things, and then those intertwine with the alpha helices, and that's where we get the overall 3D structure of proteins. And then from there, we can even build them together. We can have one protein join with another protein and form these supermolecular complexes that are also really common in the cell. So they're very dynamic. They're always moving. They're always changing. And when we figure out what they look like, that's just a moment in time, right? They are these dynamic things that change, but the way that we're able to determine their structure requires us to kind of freeze them in a certain condition and then shoot them with X-rays, and we can get what we call a crystal structure of those proteins.
What is the crystal structure? Like, what does that mean? Why is it a crystal?
So we can take proteins and we turn them into essentially a crystal. We form a lattice of proteins. And that's not something I've ever done in my work. So the specifics of what goes into it, I'm not 100% sure. But we need essentially them to be solid so that we can shoot them with these X-rays to form the crystal, to get the crystal structure. So again, that's what I mean when it's just a moment in time in this very specific environment where we need the proteins to basically almost like, I don't want to say precipitate because that's not quite the right way to describe it, but we need them to be solid enough because they're usually in a moving environment of the cell that's very full of water. So yeah, crystals.
Yeah, it's interesting how we often, I don't know, I think back to like some of my earliest training, and I know it's not really like that anymore, but this is one of the big quandaries of science communication, right? Is how do I reduce the complexity of a topic for somebody who doesn't have all of the necessary background knowledge in such a way that it is digestible, but not in such a way that it is no longer accurate, right? And so I think when we think about how we learn a lot of biology and chemistry and biochemistry and even physics when we are young, and how that translates to the real world, there's sometimes a disconnect because we think about things in almost like a crystalline form, like they're fixed in time. They're these like hard structures that are like unmoving and unchanging, but really everything's in flux and everything's just like a little bit wet and sort of falling apart all the time.
No, I think about this a lot because it is when you go through your scientific training, you're trained to think not like a human in a lot of ways. And everything has all these caveats and like, we got to split this and this. And it's like, you're always worried about being highly accurate. And that's just really not how humans think about the world. It is squishy. It is feelings. It is emotions. It is stories. And so I think that is a huge challenge for scientists to how do we maintain accuracy and also connect with people and not put them off. And there's a quote, and I don't actually know who said it, but use it a lot. Specifically, what I'm talking about, the intersection of science, communication, and the arts, is what you lose in accuracy, you gain in power. And I think it's helpful to think, what do I, the scientist, actually want the person I'm talking to walk away with? I don't really care if they know how we get crystal structures of proteins, but I want them to understand that proteins have crazy structures and we can see them. So that's the balance. And you get pushback from scientists because, like, well, actually, technically, but if I'm trying to connect with somebody and get them to buy into why they should care about science, I can't get hung up on that detail about, did I use the exact right way of describing this? Because we're like, I want the message to be powerful and for them to walk away with the same sense of awe that I did when I first learned about this, even though I had no idea about what X-ray crystallography was at that point.
Yeah, yeah, there's a huge difference between, like, the how and the why, and while the how matters, it matters to probably a much smaller percentage of the population, the ones that are actually in the lab doing the how, iterating on it, trying to improve methodology, all that good stuff, but the why is what everybody should have access to.
Yeah, everyone should have access to this type of information, and I think it is. I know we're getting a little into the weeds of like SciComm stuff, and I know there's people who disagree with me, but I think it's an ethical imperative of scientists to be doing this, and because everyone has a right to have access to this kind of information. It's makes us who we are. It dictates everything we do on a daily basis, our health, everything. So it's really important to me that we build the trust back with the people who no longer trust scientists or these big academic institutions. And that starts with meeting people where they're at and not worrying about, well, my thesis advisor would say technically that's wrong, but that's I'm trying to build trust because people don't really trust what comes out of research and science at the moment.
How do you find that balance as somebody who is, who has a lot of expectation and pressure on you to complete your postdoctoral research, to do, to be an adjunct professor and to teach and to be in the lab and to publish and all those things? How do you balance the SciComm work with the pressures of the job?
I thankfully have been able to surround myself with advisors, mentors, people who also believe the work is important. So I don't have to fight to justify why I'm taking time to do something other than run another experiment. I think that's helpful, but there's been pushback and times where I'm like, I'm sorry, this is important and I think it is. Why am I doing this why am I here being a scientist if I'm not able to go and be in my community and connect with them and share the work that I'm doing with them? I think it's wrong. All right. And if we write grants, if we write, try to get money, we have to tell what we're going to do, the broader impacts of what we're doing. It's part of a lot of funding that scientists get, and then they really don't do anything about it. So I tell people it's in my funding that I have to do this. It's part of the job.
Yeah, and that's even increasing now under the new administration, right? Like, not that we haven't always had to sort of justify why we do what we do, but now there's an added pressure to make it politically expedient, to make it like to have like almost a fair amount of persuasion about like why this matters today, right now, why we need funding right now because of this specific outcome. We can't just say we want to do science for science sake. We want to discover something new. That's like not enough anymore.
Even before this administration, that's been true, where basic science is not as fundable. It's not as encouraged because everyone wants an immediate outcome. But basic science is really what's given us so many of the innovations we have today. And I think also that's another thing that the general public doesn't really understand about science, right? They don't understand what we mean when we say basic science even, and so they're confused about where why are you studying, you know a cone snail? What does that have to, why are my tax dollars going to
Yeah.
that? And so we're doing a very poor job of explaining that as well, like why this work matters, and it's going to be the long haul till we maybe see the outcome. So instead of just funding cancer research, which we absolutely should, there's a lot of other science that needs to be done that we should be advocating for.
Yeah, yeah, definitely. It's a tough position to be in right now as, I would say, a young scientist. I can imagine that you have young colleagues who have just kind of finished their undergrad, who are starting their graduate school journey, and who are trying to sort of stay focused and center the science in their lives because there's still so much to learn and navigate, and it's so overwhelming, even just understanding how to navigate within academia. But now there's this whole added, or there has been increasingly this whole added layer and pressure of doing that within a political context.
Yeah, everyone is very stressed and very worried, and
Yeah.
I feel it as well. I don't know what will be next for me because funding is dropping left and right. It's this strange feeling of, okay, is kind of the resistance the way we fight back just to show up and keep doing our work no matter what, or do we act like this is not normal because it's not normal? That's kind of where I find myself a lot of times, is this isn't normal, why are we acting like it's normal? But also I just need to keep showing up and doing what I'm being asked to do. That's my job as a scientist, right? And I think that's kind of been the general feeling around my colleagues, both even pre-tenure professors down to graduate students and even undergraduates. Everyone's really nervous and just trying to figure out what's going to happen next. I already know several postdocs who've been let go in the past few weeks because funding has been taken, graduate offers being rescinded after being previously being accepted. So it's not a fun time to be a scientist right now. It's at any stage in your career, I can imagine.
I think one thing that maybe the public, I imagine that those listening to my podcast are relatively well aware of this, but the public at large may not, fully understand is that it's very expensive to become a scientist. It's a massive investment in time. It's a massive time and economic investment, whether it's a personal investment, like somebody is paying for relocating or paying for tuition costs or books or anything like that, but also the lab itself has to get the funding to support these researchers. And, there's a lot of training that goes into this, and it takes a long time. So you've got this massive financial and temporal investment to become a scientist. But on the other end of that, it's not like easy street per se. Yes, some scientists, and some academics are in relatively comfortable financial positions, but a lot of early career scientists are still really struggling financially. It's not like, once you check all the boxes, you're good.
Yeah. I mean,
And that's tough. Like, what do you do next, right? And I feel like there's probably a lot of people in your position where it's like, okay, once I finish the postdoc, is there a lab that I can build somewhere? Is somebody gonna hire me as a professor? Do I wanna go into industry? Do I wanna stay in academia? It's a tough time.
that these are, I think scientists aren't always viewed as full people. Our job is kind of, it's so out of touch with kind of everyday lives of people, but it is our everyday life, right? I get up in the morning and I go to work. My job just happens to be that I'm a scientist. And I don't think people realize how much this is really devastating the scientific community and a lot of the progress that's been made as far as diversity in science is now really going to be pulled back because the people who are going to make it are the ones who have that advantage. Not only because a lot of the DEI stuff is a part at the heart of a lot of these rollbacks, but um, it's going to impact small schools. It's going to impact undergraduates who are coming from primarily undergraduate institutions, which are usually serving a large minority base. Those are the people who are going to feel the effects the hardest and have the hardest time coming back from it. And I think that that's not quite being talked about enough in the scientific community. Of there's a lot of people who will be completely fine, and the people who are not going to be completely fine are the ones who are already underserved in some way in our space.
Absolutely. Yeah. 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 And I agree. I think that maybe it's because it is so overwhelming for so many people right now, but I guess the outcomes of this new and more intense iteration of tightening and of cracking down and of just honestly like a lack of respect and admiration and therefore funding for science as a whole, this is going to affect us for like generations. It's going to affect us on an international level for generations. And it's heartbreaking. And so I'm curious, how does somebody, I know we've like gotten way off topic with proteins, but we'll come back. We'll come back. We got time. How do people, or maybe I can only ask how you stay true, like keep hope, how do you show up every day? How do you not despair all day every day and like bemoan everything at work? And how do you keep sort of at least a face on that says, let's keep going, let's keep doing this?
The first thing is I'm very lucky to get to do what I do. How many people wish they could have the opportunity to be a scientist at this level? So I'm very grateful, even though there's challenges right now. That's one of the first things. I'm not grateful every day. Some days are hard because it's not always fun, but that's part of it. I also don't really take my work home with me in the evenings as much as I used to. I go home. I raise ducks. I have 13 ducks. So I go home and I sit outside and watch them uh, every evening. I disconnect from what's going on. I spend time with my kids and um, read poetry. I try to find outlets that not only they're not just distracting me, but they actually make me a better scientist to participate in some of these creative endeavors. And they bring me a lot of peace and joy and remember why I'm doing what I'm doing because it's a really creative practice for me to be a scientist. Um, and just, the it can feel like the world is falling apart. And then you go out and you kind of touch grass, as they say, and you realize it's not actually falling apart. There are things we can do and just remembering that we exist in a community that's full of other people going through similar things, even if they're not scientists, and just trying to stay as human as possible in these moments of panic. And then I can show up to my work focused and motivated because I remember that it's not just about me, it's about uh, our entire city, our entire scientific community, our entire country. And I'm no good to anybody if I'm just running in circles, stressed out all the time.
Do you feel like that is the general kind of vibe and consensus among peers and colleagues and, sort of uh, when you show up to lab?
Um, I don't know.
Or is everybody all over the place right now?
I think a lot of people are all over the place right now. People find it very odd when I say I leave my laptop at work most nights, because there's already so much stress and pressure in academia to be working and publishing all the time, right? But I just tell people, I say, no, you will feel so much better about things if you take a night off, go to a yoga class, go, do something. So I think the voices of take care of yourself are not very loud, at least in the spaces I'm in, and everyone's pretty high strung, rightly so. And so, yeah, I would just say go read a book, go do something, and then you can come back and actually do something meaningful if you're not so beside yourself when you're in your workspace.
Absolutely. Yeah, which is not always easy to
No,
do.
it's
I mean,
not.
clearly, it's yeah, and we all I think can improve on that sort of work-life balance every day. It's counter to all of the messaging that we've received our entire lives, like this sort of like deeply internalized capitalism that we all carry around with us. But it is actually the thing that will make us better at our work, right? If we can walk away from it, then we are more likely, I think, to have these like moments of insight. If we can not be burdened by it all the time, we're less likely to be burnt out and to resent our work, and we're more likely to enjoy it and recognize that how lucky we are.
Yeah, and it also is a chance for you as a scientist to get out into your community and meet people, especially if you want to be a science communication, if that's something that's important. You can't communicate to people that you don't interact with, um, in your own community. So that's another thing that's really important to me, is to make sure that I'm spending time outside of my science spaces, not with scientists, and making sure that, because then that helps me become better. If I'm trying to explain something and people are like, no, I don't understand it, then I can rework what I'm doing as well. So killing two birds with one stone in that way, I guess, taking care of myself and practicing science communication.
I love that. And so kind of to take things back a little bit as we're sort of arcing the episode itself, coming back to the work you do with proteins and to the book that you wrote with your co-author. In the subheading you say the molecular language of proteins. So of course you dive deep into what proteins are, how they're coded, what we can do with them, the chemistry behind them, the molecular biology behind them. But also you talk about the future of life. And so I'm curious about that turn, which is so often, I think, a necessary turn, but also it can be a difficult turn to say, okay, this is what we know. This is how we know what we know. This is what we're actively doing with what we know. But wow, look at all of the potential. So how did you guys, I guess, decide how does one become a semi-futurist in their field and decide what to focus on and what is science fiction and what is potentially going to be science fact?
Yeah, that's a great question that I've never really been asked before. Such a great question. Yeah, I think, again, the best scientists and the ones who've been the most innovative and come up with the craziest ideas have been the ones that were just playing. They're really just exploring and coming from a place of, I want to know more, not I want to invent the next big thing. In the book, we talk about the one of my personal favorite scientists, Linus Pauling, who actually was the one who discovered that these proteins have these helical shapes. And he was just in bed sick one day and was just messing around and figured it out, right? And it changed, you know, an entire field. So I think if you're trying to be a futurist, I think you might be doing it a little bit wrong, because then you're already narrowing down what you think should be done. Not to say that you shouldn't have a plan or a goal and it's not important, but that's why I love the Nobel Prizes every year, because most of them were just starting out as scientists when they made whatever discovery it was, and it was sometimes by accident, or they were just paying attention at the right time and were curious enough. So I think that would just be my take on this idea of being futuristic about your field, and specifically in the world of proteins. The reason we do kind of talk about it as the future of life is because of this idea of protein engineering, that we have figured out how to change biology in a way that we've never been able to change it before. And that discovery, one of the ways we do that is CRISPR, which also won a Nobel Prize not too long ago. But again, that was discovered just kind of by chance. That was not what they were trying to do with anything, was develop this tool that's going to change healthcare, right? Um,
Absolutely. And I love the point that you made too, that like when we discover these things, sometimes by chance, sometimes it's obvious, like, oh wow, look at what we just cracked, or look at what we just stumbled on. What are the implications? But very often it's like, ooh, I think I messed up
yes,
here. Like, this can't be
Yes.
right. Like, everybody was saying it was going to be this way, and now it's this way. What did I do wrong?
Exactly.
And it takes a while to figure out that the wrong thing has a lot of right implications.
Yeah, I teach an organic chemistry lab, and every semester something obviously goes terribly wrong with some student. It's not supposed to be this color. I'm like, well, maybe it is. Maybe you are just discovered the next Nobel Prize winning reaction. Like, you don't know that this is the wrong thing. And also, it's a point that my co-author makes a lot as well when we're talking about this is as a scientist, you have to make the right mistakes. It's not about not making the mistakes. What are the right mistakes to make? What are the right risks to take? And in this era of our funding and all of that, it's a little bit scary to take risks. But I think even before that, we were veering away from taking some big risks, some big swings, and even something preposterous. Even Linus Pauling got a lot of things wrong. He was completely wrong about the structure of DNA. So we can't be afraid to take some big swings as we're allowed to, whatever confines we have with our research situations. But we have to maintain this humanistic approach to science, that this is play, this is curiosity driven, this is not should not always be just a problem solving initiative. I think that's the step that comes after exploring and being creative with your science.
I love that. I'm so glad that you made that point, and I'm so glad that that is sort of where this conversation went, because fundamentally, I'm with you 100% of the way, and I think that that is um, the aspect of these conversations that's often either buried, and I think it's implied, but for a lot of people, it's actually not implied. So I'm glad that you brought that out into the daylight. I want to make sure, because I would be remiss if I didn't ask you as we're kind of finishing up. I do this a lot. Everybody listening to the show knows that I do this a lot. There's a lot in the book that we did not even remotely get close to. And that's okay, because the whole point of the show is not to read the book out loud to you. It's so that you get, your appetite whetted, and you go buy the book and you learn on your own about these things. But did we skip anything super important? Like, is there anything that you're like, oh God, I should probably say this, or like my co-author is going to be mad?
No, I think the only thing I would say is the number one question I've gotten asked as this book has, been coming into people's hands is, am I going to understand it? Is it for me? Am I too dumb to read this? And the whole point of this book from our perspective, from Shahir and I's perspective, is so that we can give this to our moms, our electricians, the soccer moms that I go see every week. This is for everybody. You might not understand every single thing in it, but we really did our best to make this accessible and approachable. And our goal is to spark curiosity. And we wanted to make it personal. We share a lot of our own personal stories in this as well. No, you can read it. It's okay. You're not too stupid to read this book. It is for you. You will understand it. And then hopefully that leads people to feel empowered to seek out good information, because they were treated with respect and curiosity, and got a little taste of something that maybe they thought was not accessible to them. So that's the only thing that I think Shahir would be upset if I didn't say, is this should be accessible for everybody, and we hope that it is.
I love that. And I think it speaks to one of the sort of central tenets that I often talk about when I personally am teaching SciComm, which is that you should never underestimate the intelligence of your audience, but always underestimate their vocabulary, right? There are so many things that we can understand conceptually that maybe the reason we get lost in the weeds or we get scared is because the lexicon is overwhelming to us. It's like, oh, I'm never going to understand this. Listen to all those big words that you guys keep using. And it's like, there's a reason that anybody in any trade uses shorthand and uses specialized language. It makes it so they don't have to say 50 words when they could only say 2. But we're okay using 50 words when we're talking to somebody outside of the field. That matters. And I'm glad that you made that point.
Yeah, absolutely.
Yeah.
Yeah.
Conceptually, this stuff is not too complicated for people to understand. It's not at all. Logistically, sometimes it gets really complicated, but that stuff doesn't matter as much.
Yeah, and I appreciate what you said too. We shouldn't, as scientists, underestimate the intelligence of the people who haven't had the same scientific training that we've had, right? It really does a disservice to what we are trying to do as scientists, which is serve our communities and make the world a better place. People are smart, and if you spend some time trying to explain something, they will understand it. Turns out it's not actually that difficult to understand some of the stuff that we do as scientists. We're not as smart as we think we are. We just memorized a lot of.
And that's exactly what you did.
Yeah. That was.
Yeah, you took the time. You took the time in this book to explain a lot of this stuff. And now people who are smart can pick this book up and they can say, oh, okay, I get it. Like that makes a lot more sense now.
Yeah. And hopefully empowers people, especially moving forward in this crazy time for scientists and everything, to help people find the right information because somebody talked kindly to them about something complicated.
I love it. I love that. Thank you so, so much. Thank you for taking the time to write the book. Thank you for taking the time with us today to share some of these insights and to not just talk about the content, but also to get a little bit meta and talk about the why of it all and some of these additional layers of that I just think that are really important, especially given the context that we're in right now, the political landscape that we're in right now. I'm just really appreciative for you to lend us so much of your time, especially when you have, I'm sure, lots to do in the lab. So Maggie, thank you so much for being here today.
Thank you so much for having me. I really appreciate the opportunity to talk about this book and talk about science communication and what we can be doing to help repair the problems that science has created in the past little bit. So I appreciate you letting me talk about something I'm very passionate about.
Absolutely. Everybody, the book is The Color of North, The Molecular Language of Proteins and the Future of Life by Doctors Shahir Risk and Maggie Fink. Everybody listening, thank you for coming back week after week after week. I'm really looking forward to the next time we all get together to talk nerdy.